Hi everyone, welcome to my channel Pure Biology, and today's topic is the function of antibody or immunoglobulin, and this is another video related to immunology. And here I just explain you what are the functions main of the antibody or the immunoglobulin. And before going to this topic, I want to say thank you to all of you who have subscribed my channel, and the newcomers obviously share this channel with your friends if this is helpful for you. And I'm trying to give the knowledges that related to the biological science, so you just continue with my channel and try to share it. And obviously who have already subscribed my channel, so please you press the bell icon for all notifications. Okay, so here first the structure, that is the oxonization. What is the oxonization? Oxonization is actually it's the recognition of the foreign particles and the phagocytosis of the foreign particles. This is done by this process, oxonization. Okay, and this phagocytosis process is done by the macrophages and the neutrophils. Okay, macrophages and the neutrophils. And here actually the FC, and by this way it can maintain the defense system. And here the FC receptor, generally FC receptor, that can bind the constant region of most IgG molecule. Okay, IgG molecule is one molecule. Obviously it is antibody molecules. There are different types of antibody molecules are there, and it is one of them. So here we have taken this IgG molecule, and there receptor, this FC receptor, that can bind with the constant region. Okay, and generally the interaction between the single, in case of single FCR and FC region. FCR means receptor, FC receptor. Okay, so FC receptors generally bind with the FC region of IgG molecule in case of single. Okay, so first what we have known here, that the FC receptor is present in the IgG molecules. Okay, FC receptors are present in the IgG molecules, and the binding of FC receptors and the FC region is very weak. But in case of single molecule, but when 2, 3 or 4 molecules of the antibody is there, in that situation the binding become very strength. Okay, the binding become very strength, and this binding is occurring with the, if this binding is occurring with the bacterial cells, that means first bacterial cell is recognized, this is the foreign particles, and after recognition they totally bind, they are all the antibody, these IgG molecules of this antibody, they totally bind to this bacterial cell or the foreign particle. And what is the function after this, after the recognition of this bacterial cell? They engulf, or phagocytosis is possible here, and by this way they try to they totally damage the foreign particles, and this is the process oxonization. And in this oxonization process, the two cells, that are the neutrophils and the macrophages, both help in this oxonization process or phagocytosis process. Okay, the second one is the antibody dependent cell mediated cytotoxicity. What is the antibody dependent cell mediated cytotoxicity? Here the linking of antibody bound to target cells. Okay, this bound to target cells with the FC receptor of the NK cell. Here it is possible by the FC receptor, and here also it is possible by the FC receptor. And NK cells means natural killer cells. And what are the cells here? Here the macrophages and the neutrophils. And here the cells are the NK cells or the natural killer cells. Okay, and they can kill the cell. and that causes the death of the death by apoptosis. Apoptosis is another process by which they damage the target cell. Clear? But here the movement of the antibody is very necessary, because and generally here in this process the movement of the antibody is not possible. Actually the target cell is first recognized, because this target cell generally bind to this either surface of the macrophages and phagocytosis, so they are already present, these Fc receptors, and its function is to recognize this and damage it. But here the movement of the antibody is very important. Why? Because first they recognize, then move towards that place, and after this the apoptosis process or damaging of the cell is occurring. Clear? So here the delivery of the antibody to the mucosal surface is that requires the movement of the immunoglobulin across the epithelial layers. Clear? So antibody, delivery of the antibody, if immunoglobulin, that the possibility of the immunoglobulin, the movement is possible. In case the movement is possible, then the recognition and damaging of the foreign cell is possible. Clear? So here the capacity obviously depend on the properties of the constant region. If the constant region is more powerful, then they can easily transport from one place to another place. But if the constant region is not more powerful, in that case the movement is possible, but very time necessity is occurring. Okay. And here in that situation, one example is very common, that is the humans. In case of human, this transfer the IgG molecule from mother to fetus, and this is possible during the trimester gestation period. Trimester gestation period is, that means 28 to 40 week of pregnancy. Clear? 28 to 40 weeks of pregnancy. In that situation, the antibody must be transported across the placental tissue. What is the function of the placental tissue? That actually separate the mother and the fetus part. Okay. So what is the function of this IgG molecule? They have to cross this placental tissue and then protects the fetus from the pathogen. Okay, if its capacity not occurring to cross the placental tissue, then in that situation they cannot protect the fetus from the pathogen. Clear? So here the movement is very important. Here just recognize and damage, and here the movement, recognition and damage, or you can say recognition, movement and damage. Okay, so these are the two important functions. One is the ADCC and another is the opsonization. Two processes are very important, that are the main important function of the immunoglobulin. Clear? Next one is the complement system. Okay, so next is the complement activation. What is the complement activation? First, we have to know that complement is what? Complement is actually plasma protein, not a single plasma protein, group of plasma proteins. Okay, group of plasma proteins are there in the complement system. So generally, what is the role of the complement system? Actually, complement activation is the series of enzymatic events, and that leading to the generation of specific complement components, and that causes opsonization or the phagocytosis. So what is happening here? In case of complement activation, not a single protein, a group of plasma proteins involved here, and here a series of enzymatic activation is occurring, and these enzymatic activations occurring to produce a specific complement component, and this specific complement that causes the opsonization or the phagocytosis of the foreign particle. And here this is seen in IgM, or in case of human, obviously IgG immunoglobulin. Okay, and here the I just mentioned here 7 glycoproteins are there. 7 glycoproteins are there, which are known as the complement system. Okay, how much glycoproteins are there? 7. Okay, 7 glycoproteins are there. So one important there is another one example is just explained here. One byproduct, that is, suppose in the complement activation situation, the important byproduct is C3b or the complement 3b. That is a protein. Obviously this is a protein fragment, and the production of this protein fragment that induce to binding with the non-specifically with cell. Okay, not specificity is necessary. That means it attach only the bacterial cell, it attach with the virus cell. No, that means this is the, suppose this is the byproduct, one byproduct, that this is very important byproduct of the complement system, that helps to bind non-specifically cell. And this binding is what is creating, by this binding, antigen antibody complex form just near the site of binding of the cell. Okay, and this induces actually the opsonization and the phagocytosis of this foreign particle. So opsonization and the ADCC, obviously, those are very important. In one case, the immunoglobulin just remain in substratum way, or these are the cells where this is possible if the foreign particle just close to these cells, this is possible, or the movement of the immunoglobulin occurring in case of ADCC. Okay, so in this oxidization process, the cells are very important because the surface of this cell FC receptor is there. Okay, and this FC receptor is very functional because the foreign particles actually recognized by these receptors of constant region, and then the damages of the foreign particle is possible. So FC receptor is very important here. And in complement activation process, some byproduct is very important, that is actually induce the processing of the opsonization or the phagocytosis of the foreign particle. And in case of ADCC is what was there, that the immunoglobulin just move from one place to another place and just recognize there the cell, and then engulf or phagocytosis or damaging of that cell, or apoptosis is the main important process by which they can damage the cell. Okay, so there are the three important processes we have learned now. The function of immunoglobulin, or how the immunoglobulin act to destroy the foreign particles or maintain the defense system in the immunity. So it's done. This explanation is done here. So just you read with the after just watching this explanation, then you consult any immunology book, then you can easily understand. Okay, so thank you.